Thanks, Ellen. So I get to do everything to introduce myself. So I'm Paul Eder. I do the Phase 1 program here, and that's probably all they ever ask me to talk about these days. But I'll give you some idea about what has changed in early clinical trials. And throughout the year, you hear from people talking to you about the latest innovations in cancer treatment. We're a cancer treatment center that tries to bring improved cancer metrics to patients. The other thing you hear about is these exciting new targets that might eventually become drug targets to actually improve the care and the health of patients with cancer. That huge gap between an idea and some initial experimental work and a drug that any physician treating cancer patients or any provider treating cancer patients can prescribe is a huge gulf, and that's really what early clinical trials are. And the beginning of that are really what we call Phase 1 clinical trials. So in the past, we had a model where drugs were discovered, drugs were in very unusual ways brought into the compendium of trying to treat cancer, and then we treated patients. And whatever we saw, we used that to help us generate the next generation of trials. And if the drug looked promising, then we figured out why it worked. It didn't have to know that before we started. Things have changed. Things have changed dramatically. We still discover a lot of drugs empirically, but increasingly, we're trying to determine what the molecular profile is of a particular patient based on all the new biology that you've heard about here and elsewhere, and how can we apply that to actually make a specific cancer treatment for a patient, a specific patient. So in the past, this is sort of the overlapping Venn diagrams. And what you'll notice about what's changed here is not that these things are different. I mean, toxicity, what we call pharmacokinetics and pharmacodynamics are still there at the same, but we've added many, many more components to this, which adds to complexity, cost, and unfortunately, despite our best intentions, sometimes to time. So the future that we're looking at for patients with cancer is going to involve something more like this. When patients are referred, increasingly we're going to try and looking at their archived tumor material, try and determine what it actually is that drives their tumor, what makes their tumor behave, things that Ellen was talking about, the genetic screening. These are some of the techniques we use to describe these, all of which are actually in clinical trials at Yale now. Then a patient's allocated to a trial based on these molecular characteristics. Less important in the future will be whether the tumor originated in a particular organ system, but what is it that makes this cancer behave the way it does? And how do we monitor patients? Here's the clinical monitoring that we look for. The tumor shrunk. This is what patients ask. This is what we look for. And despite a lot of elegant biology, this often determines whether or not the patient's going to survive longer as a result of treatment. But as we do these studies, there are a compendium of other things we need to check as we do this. Try and determine, is our treatment benefiting patients? And if it is, we can learn this and apply this backwards to other patients now, rather than necessarily waiting a particularly long period of time to do this. And this monitoring has changed to the extent that you've heard descriptions about, well, when patients' tumors progress, we'll reanalyze their tumor and figure out why it was resistant. Very important, I think, for us is to also understand that we have to reanalyze these tumors when they actually respond. We pick one or two characteristics out of 40,000 possible proteins, exomes, or protein coding exomes, and we figure, well, it must be this one because this is what we thought. Increasingly, we have to be sure that, in fact, when we do see disparate results, when we see a 30% response rate, when patients have a target, well, what's about the other 70%? What didn't we know? And this is a way to try and help us do this. Here's an example of looking at serial biopsies over time. This is not a patient who relapsed. This is a patient who actually responded. And the slide is truncated a bit, but we did this over 6 cycles of treatment to show that over time, our target was still being engaged. Ultimately, of course, the patient demonstrated resistance. We had to go back to this approach. But these are the kind of things that we're adding to cancer clinical trials now, with the consequence that trials are getting increasingly complicated and, as I said, characteristically expensive as well. So sort of our favorite tool for talking about these is a biomarker. And this is the FDA's point. The answer is basically this is something that tells us about a process, normal or pathologic, and the pharmacologic response to our treatment. And it helps us evaluate that treatment. There are types of biomarkers, and they can be categorized in many different ways, as you can imagine, but probably an important way to look at them is the following. They're what we call prognostic biomarkers. These tell you something about the course of the disease at diagnosis or at any point. Sometimes it's a data set, like a Gleason score in prostate cancer, gene profiling in breast cancer, or a single point. Is there estrogen receptor expressed on a breast cancer tumor cell? Is there a mutation in non-small cell lung cancer in the epidermal growth factor gene? And they have these predictive biomarkers. And when you hear about personalized medicine, this is largely what we talk about. Is there something about a patient that tells us he or she will respond to a specific therapy, like HER2 new expression in breast cancer or EGFR expression? Something that's to be understood is that increasingly we're beginning to understand that many of what we call the standard of care drugs, the basis on which we add additional drugs, there are now predictive factors for these drugs as well. And in small clinical trials, when you don't take this into account, you can get very different results when you do larger trials if you haven't segregated these patients prospectively. And these are just some of the examples, such as methylguanine transferase in patients with glioblastoma or TOP2A in patients with breast cancer. And lastly, what I would call pharmacodynamic biomarkers. They tell us whether the specific treatment is affecting its target. What are the consequences of that? These are just some examples that we've used in the past. And so we take these biomarkers and we can use them to help select the appropriate therapy for a patient. For a group of patients, we can determine what's the minimally biologically effective dose. I think it's kind of a truism that the less drug you give, the better. But it's also important to realize that for almost all therapies, variability in humans and how they handle this, the pharmacokinetics I'll talk about, means that there's going to be a wide range of what each human being will have after receiving a particular dose of a drug. And we need to know what's the minimum we need to do, but what's the optimal dose for a patient population, where the variability may be 30 to 40% from one patient to another. These biomarkers help us select an optimal dose and schedule. These can tell us how long a drug persists in somebody's bloodstream, how high we can go to get these two levels, help us to make scientifically based go, no-go decisions. It's another truism of drug development is you don't want to spend time on drugs that aren't going to be successful. The sooner you know this, it allows you to allocate your resources towards those drugs that have more potential. So quickly determining a drug has not got the potential to really be a therapy is very important. Helps us to take increasingly limited resources and invest them into areas where there's a greater chance for benefit. And so increasingly with biomarkers, we try to get as early indications we can about the drug. What we call, does the drug actually interact with the target? Are we able to get enough drug in frequently enough that we're able to actually show that we engage the target? And when we did engage the target, was there a biologic consequence? Was our theory right about why this target was important? And most importantly for patients and everybody down the stream, on the biological consequences of this target interaction producing the desired effect, which means a proof of concept. We're actually making the tumor regress or at least stabilize. So pharmacokinetics, in summary, is basically what the body does to the drug. This is the Latin grammar of clinical trials. If you can't do this right, you never get to the second step. I guess for those who don't study Latin anymore, but at one point, this is the entree. And we call this the so-called ADME. So is a drug absorbed from its site of administration? Easy to do intravenously, which has characteristically been what we've done in oncology, but increasingly as we go to oral drugs and others, this becomes a more important factor. Once a drug's taken up into the systemic circulation, it's distributed throughout the body. And we usually only measure the drug that's in the bloodstream. And one of the underappreciated areas has been how much of that drug is actually potential to interact with the target because it's not protein bound. Most human drugs are at least 95% bound, which we consider a good drug. Many are 99%. So you've got to fill up all of these extra spots before you actually have drug that's able to interact with the target. And sometimes that protein target in the bloodstream, which is usually albumin, may be mimicked by other sinks in other tissues. So it's very important for us to actually know how much drug we're getting to a patient that might potentially interact with its target. No drug stays around unchanged. Metabolism is the process of changing drugs into inactive compounds, but for many of them, there's an intermediate step when these metabolites may be actually biologically active. We use this for some drugs like cyclophosphamide and such, where this markedly reduces the toxicity of the drug because it comes as a prodrug. It only gets activated once it's in the bloodstream. And eventually, the drug and its target metabolites are all eliminated. And all of these things go into us helping better understand what happens to the drug when it's given to human beings. And so this is the typical PK sign of what we call drug is given intravenously, the drug distributes, and then it's eliminated based on its metabolism, excretion, etc. But this is great from the point of view of understanding drugs, but when trying to understand drugs in the clinical context, we then have to add other variables to this. Here's an example of an oral drug, for instance, but here's the minimal effective concentration. So all this drug below here doesn't contribute to the effect we're looking for. Only during this period of time do we actually see this. This helps us determine how frequently we should dose patients, what happens if the drug begins to accumulate over time, and it looks like we've got a nice therapeutic margin between the toxic levels here, but as we give more drug, if we give it too frequently, we may exceed this. And given human variability, this range needs to be as broad as possible to minimize toxicity for special groups, but I'll talk to you further about it. This little aphorism at the bottom, I think, is actually fairly succinct, talking about the relationship between a dose-response information. This also happens to show up on multiple places on the FDA website, so clearly other people who think about this think this is pretty important. And so, again, the relationship between concentration that was the wrong word, and drug effects are what we call the therapeutic index. This is the response we're looking for. This is the toxicity we don't want. There are some drugs where we use very, very small concentrations of highly potent drugs. There are other agents like Tylenol, which you take for a headache, where you give gram doses of it. It doesn't matter that the drug's relatively inactive compared to its dosage form. The fact is it's got a huge therapeutic index, and that's more important than actually the molar potency. It's a general rule that the more potent drug is on its target, the fewer side effects, the greater the therapeutic margin, but it's not always true. And again, it's important that any drug that's going to be given to patients and be approved by the FDA, you've got to know this relationship between the dose, the desired effect, and the undesired effects. Without that, they're really concerned about you would know how to safely do this in humans. So the second concept, besides pharmacokinetics, is what we call pharmacodynamics. What the drug does to the body. If you're taking an antihypertensive, it's lowering blood pressure. If you're taking an anti-glycemic drug, you're lowering blood sugar. In cancer, it's a much more subtle one, and what we're looking for is actually tumor regression or tumor stabilization. So plasma concentrations are sometimes a surrogate for this. In animal models, we're able to determine what dose produces a desired effect in a model system, and this helps us link this to humans, getting us an idea of what exposure we need. Since most animal systems are highly idealized, if you can't reach the concentrations in an ideal circumstance, you're very unlikely to see benefit in the more complex patient setting where multiple factors are involved. And so sometimes we use this as our initial target for where we're going to go with the drug if we can only achieve those levels. And so it's important to remember that these support our choices of dose and the interpretation of the biological effect studies and also for drug-drug interactions. Very few human beings, unlike Jackson lab rats, are taking only the drug you give them. They're on many other agents that they need, sometimes for their cancer, sometimes for other diseases. Got to keep all of that stuff into context. So we've used pharmacokinetics as a surrogate for this pharmacodynamic endpoint, looking at these areas like total drug exposure, the concentration in the bloodstream at steady state. You may have seen patients, I'm sure you have, who've been taking anti-epileptic drugs. You measure the levels because there's a threshold below which patients are at risk for epilepsy. Too high, there's a risk for toxicity. We don't often do this in medical oncology, but it's very, very important. The transplant people know this about the injection meds that patients get on the 7th floor and the 11th floor. Again, other factors that go into this. So these PD endpoints have been very helpful more often for determining toxicity than efficacy. But some of us remember back in the beginning, at least the stories we heard, about the first treatments for acute lymphocytic leukemia in children. Children give a combination of drugs like methotrexate and mercaptopurine, had sort of one of three outcomes. One is they got significant toxicity, usually bone marrow suppression. You had to decrease the doses of the drug. A second group of patients got no effect, and that's great. When they come back to clinic, there's no side effects, it's easy. And there's the third group that had some drop in their counts, not too dangerous, but some drop. And when you looked at the outcome for these groups, you found that those people in the middle were the ones who did the best because they had enough drug to get biological effect, not enough that you had to decrease the dose or run the risk of underdosing them. Those patients who had no toxicity also were getting much greater clearance of the drug and much less activity. This we found out 30 years later when the pharmacokinetics and pharmacodynamics could actually be explained. So we've used this kind of clinical toxicity to help us determine, in fact, what the proper dose should be. Do we change the dose because of neutropenia or thrombocytopenia? Other agents, you've seen all these. So we know whether it's a rash, you've got to decrease the dose of sorafenib. Patients have had diarrhea, so irinotecan has to be decreased. We use these sort of pharmacodynamic endpoints of normal tissues to help guide drug dosage and help us drive drug scheduling for patients with cancer. And here's an example of how this has been used. As the plasma concentration of Paclitaxel increases above this threshold minimum, this increases the amount of neutropenia you see. So you try to say, well, 50 or 60% neutropenia is not so bad. Maybe here's the dose that we want to be at. There's a greater chance of showing some benefit as opposed to, yeah, it'll be great, nice and safe down here, but we're not likely to benefit patients if we stay at a level that's too low to actually get clinical benefit. So pharmacodynamics has also moved into the exciting realm of what we call molecular targeted analysis. These are examples of just looking at phosphorylation of certain target proteins. These are the kind of things that show up in all of the journal articles. These are the kinds that any clinician knows if he or she can get a couple of these into the paper, it's going to get a much higher profile of where it's going to be published than if it's just a black and white page with a few tables. Now the question will be, does this really tell us anything? If these assays that we're looking at, these antibodies are not well validated, if these targets are not well validated, we get a lot of pretty pictures and no real information, and sometimes misinformation, because we assume we've accomplished something when in fact we haven't. But very importantly for us is to try and find out if the target is phosphorylation. Diminishing that phosphorylation at a certain dose can help guide us into whether or not this is an appropriate dose and appropriate patient population to treat. Now this is Vemurafenib. I guess it's called Zalboraf, is its trade name. This is a drug that was introduced several years ago for the treatment of patients with malignant melanoma with a mutation in the so-called BRAF gene. In this group of patients, you can see dramatic benefit.
Has joined the conference.
Sometimes this is demonstrated on something as simple as the grayscale of computed tomography. This is a great bioassay, a great biomarker, because everybody can get it. It's relatively easy to repeat. Much more complicated is FDG PETs, just because they are more expensive and more time consuming. But again, showing you that for a specific concentration of the drug, you can actually see these kind of clinical...
Has left the conference.
And in this particular trial, looking at some patients who were able to be biopsied, the brown shows the activity of the target protein at the time you started. When you see no longer that activity, it's an indication the drug's doing what it's supposed to do, that it's inhibiting tumor proliferation and downstream markers of proliferation. So this is an example where this helped lead us to the next slide, which actually shows you that in fact those elegant pharmacodynamic endpoints correlate with changes in the size of the tumor per patient, and just as importantly, how long the patient remains on study. There are many factors that go into this. For many of the relapsed patients on this study, their expected survival might range somewhere about 4 or 5 months perhaps, and what you can see is that there's a substantial number of patients who get real benefit from this. So in a trial that needs to accrue hundreds of patients, it may take years to get this data done. If you can in fact show this earlier in these kind of assays, you can presume two things. One is that the likelihood the trial is going to be a benefit is greater. And secondly, we should pursue this idea rather than just waiting to the end. 2 to 3 years makes a lot of difference for patients with cancer. And changes in tumor volume are very sensitive because you can see them over a wider dynamic range than certain other assays. And again, these are often relatively repeatable compared to tumor biopsies and such. And these are examples showing you what were called so-called RECIST criteria, which is a normalized criteria for around the world for interpreting patient tumor shrinkage, the so-called World Health Organization, which requires 2 dimensions, and here's 3 dimensions. This kind of information is actually acquired. It's not easy because it requires additional interpretation, additional analysis by computer programs, but this is a much more sensitive measure of whether or not the drug's actually benefiting. And in early clinical trials, you're not necessarily trying to convince the FDA that this is an approved therapy. You're trying to demonstrate to yourself and whoever's behind this that this is a good idea, that you've picked the right patients and this drug can work itself or in combination. So these kind of things sometimes are more valuable for us in early trials than they may be for somebody who's looking at late-stage development. PET scanning has emerged, as you saw from that first slide about Vemurafenib, as an important means for looking at tumor response. And here's an example outside of oncology. This is an example of psychiatry, looking at occupancy of a receptor. So you can give a very small amount of a radiolabeled target on a receptor in the brain, and you can actually show that about 80% occupancy is associated with the best clinical responses you're going to see. And so you can dose patients and get an idea for what's the appropriate dose for patients. So instead of a phase 1 trial that might take 2 to 3 years, you can get this done in 12 patients. And with antibodies, this is much easier to do than it might have been in the past. So we can determine very quickly the appropriate dose, the appropriate schedule for patients, and actually help us save a great deal of time in the development and a great deal of patients being treated with suboptimal or potentially therapeutically excessive doses. Here's another way looking at magnetic resonance imaging. So this is an example of a drug that targets tumor angiogenesis or blood flow. And just looking at these crude reconstructions here, they're not crude,
Has left the conference.
reconstructions, you can see there's often not very much change in the actual size here. Not enough in that volume slide or that one dimension to somebody say this was a response. But if this is the intense blood vessel growth and metabolic activity at the beginning, over time, you can see the tumor becomes increasingly hypovascular, requires and has less and less blood flow. And so even though there's not a dramatic change, we can tell in relatively 2 months that these patients are deriving benefit. Our hypothesis at least merits additional study, helps us to determine whether or not the drug's actually accomplishing its target. Done without biopsying patients, done without some of the imprecision that comes with using individual laboratory techniques for antibody analysis. An MRI technique is something that can be published and done at many sites, so it doesn't require the difficulty of having things done at central repositories. Again, this is the kind of test that we can do on everybody, not a selected number of patients with biopsiable tumor, to help us determine whether in fact our hypothesis is at least correct that the drug does in fact inhibit tumor blood vessel growth. So another important role for biomarkers is helping us predict the therapeutic targets. Okay? Which of the drugs patients should actually get with an attempt to try and improve their clinical circumstances. So translocations or mutations have been the most important biological targets for us, where the therapies have been most effective. BCR-ABL in patients with CML was really the first contemporary targeted agent. Others, such as the work in lung cancer that Tom Lynch, others particularly pioneered, and others, showing you that when we find these translocations or mutations in patients, these can have an extremely high predictive value. In early stage patients with CML, who get imatinib or one of its successor drugs, response rates are 95+%. In patients with EML4 alpha mutant lung cancer, it's about 80%, and in patients with EGFR mutations, it can be 90%. So patients get drugs with a high probability of benefiting them, and that's clearly what everybody's interested in. Gene amplifications have been predictive for patients benefiting from drugs like trastuzumab in breast cancer. Other areas where it's being looked at in FGFR and MET genes. Overexpression has been a bit of a misleading for us, because an initial hypothesis for the treatment of patients with drugs like gefitinib was that EGFR expression alone would determine which patient should benefit. We now know that it's not that. It's this mutation that was unappreciated at the time the drugs went in the clinic that determines sensitivity. Expression of a gene in human tumors has likewise often not been terribly effective in predicting patients, although there may be a change here. Certainly in patients with lymphomas, expression of CD20, if they're to get rituximab, makes an enormous difference in the likely benefit. You don't see us routinely checking this because about 85 to 90% of human tumors actually express this anyway. So you don't need to look for it given that probability. But as other drugs come along trying to mimic that, we're going to want to be able to better select those patients. And recent work looking at phosphorylation of the MET oncogene has selected patients with non-small cell lung cancer into 2 groups. Those who derive no benefit at all, and those who derive an enormous benefit. So again, as we get more sophisticated, we apply better rigor to looking at these other things like expression, overexpression, which up till now haven't been as helpful as translocations and mutations. We may be actually getting to the point that these become a more routine part of what we do. And certainly looking at loss of function genes like the BRCA genes in breast cancer can make an important difference in selecting patients for therapy. So here's a slide just showing you what happens with gefitinib as first-line therapy in mutant patients with non-small cell lung cancer. So here's patients selected for either treatment with gefitinib or with standard chemotherapy. You can see that the complete response rate and overall response rate in this particular trial is 3 times greater. Although there's toxicity of gefitinib, it's a fraction of the toxicity of platinum-based chemotherapy. And looking at the progression-free survival for these patients shows a substantial difference. This particular trial showed no survival benefit because, again, EGFR is predictive for response to gefitinib. It's also a prognostic factor. It's much better to have that mutation than to have other mutations such as RAS mutations or be in that 50% of lung cancer patients that we're not quite sure what drives their tumors. So you have to be careful when you interpret these data, is to walk away and say, well, we've already changed the natural history of lung cancer here. It's much more subtle to do that. It takes a little longer. Another example has been what we call synergistic cytotoxicity. And here's a circumstance where conditional lethality is the term I was looking for, in so-called PARP1. PARP1 inhibits DNA repair. In most humans and in normal cells, there are multiple pathways for DNA repair. And one of the most common is homologous recombination, and this double-strand DNA breaks, which are the most lethal and most dangerous types, induced by radiation, chemotherapy drugs, and can occur spontaneously. In those patients, this HR pathway repairs these, cells survive and are unchanged from their genetic program. In patients with BRCA mutant breast cancer, this gene is lacking. And as a consequence, there are backup mechanisms. These mechanisms are effective. They're not perfect, and they're not as error-free as homologous recombination. But a drug like olaparib that targets PARP, which is very important in this secondary pathway, will leave normal cells alone because they have and prefer this homologous recombination pathway, whereas the BRCA-deficient patients, they don't have this. So they're forced to use this pathway. As a consequence, there's an enormous therapeutic margin with this. Here's an example of an olaparib trial showing tumor regressions, of course, showing decreases in tumor markers, and showing the duration of time on study. So patients who have responses stay on study longer. They stay on study much longer time than patients who don't respond. And once again, in these assays that we're looking at, our ability to be completely predictive is not anywhere near 100% yet, partly because our assays may be incomplete, almost certainly because the biology is a little more complex than we can accept that. So this so-called proof of concept, again, much like the example of showing the EGFR mutation, breast cancer BRCA mutations are a very bad thing to have in breast cancer, but if you have to have breast cancer, you're going to live longer if you're one of these patients. And so just looking at this doesn't mean we've changed the survival of these patients. Those studies take much longer and are much more subtle to do. In addition to just selecting the appropriate patients for treatment with the drug, we can also use molecular genetics to help us determine another route of personalized medicine, and that is using pharmacogenomics to select doses for patients. Here's an example of what happens to an intestine treated with a drug like irinotecan. Here's the normal intestine. Here's what happens when you get an excessive dose of irinotecan. The loss of all of the surface area means there's less reabsorption of water and nutrients. Patients can present with debilitating, even lethal diarrhea. Over time, you stop the drug, it recovers. Here's an example of a patient who has an infiltrate in his or her lung because of treatment with gefitinib. So 1 to 2% of patients who get treated with gefitinib develop an interstitial pneumonitis. It'd be great for us to know who those patients are, so that, yes, we can benefit patients, but also know which ones we have to be careful of. And here's another example looking at what happens with drugs like Surrogate. So if we're able to predict what the toxicity of a specific drug is at a specific exposure, it helps us customize the dose, personalize the dose for that individual patient. And this genetic variability in cancer drug metabolism is very important in the drugs like irinotecan, where the toxic event can be eliminated if you exclude patients who have specific mutations that prevent the rapid elimination from the drug in an appropriate manner. Tamoxifen is a drug with a relatively good therapeutic index, but tamoxifen is a prodrug. Unless it gets activated to what's called endoxifen, the drug interacts much less well with its target. So as a consequence, patients with specific mutations in drug metabolizing enzymes are at risk for actually getting an ineffective dose of the drug, and as a consequence, deriving no therapeutic benefit. There are many other agents in this class that can be used, and knowing which drug to use would make a difference. Examples of this are thiopurine methyltransferase, 6-mercaptopurine metabolism. Patients with a mutation in this gene get about a 4-fold exposure. So we don't have to not treat them. We just give them 1/4 of the dose, and we get the desired exposure that we want. Again, with endoxifen, you change the dose for people who have mutations in 2D6. You decrease the dose by about 1/4 in patients who have this mutation in irinotecan. Most drugs will have multiple mechanisms of metabolism, some of the preferred ones, and methotrexate. So we don't use these to eliminate patients from treatment. We use this to customize the appropriate dose to get the desired therapeutic effect without excessive toxicity. So these are the kind of things that we can take advantage of in our personalized medicine approach. And so There are a lot of steps in drug clearance where genetic variation can affect this. It can check the activity or the number of these genes that are responsible for drug elimination, the enzymatic addition to increase the water solubility for further elimination. This is just a list of things to let you know that there are about 85 or more genes that have been described that play an important role in drug elimination. Not in every drug and not in every patient, but if we can characterize these, we can better understand what the appropriate dosing for an individual patient is once we've selected what drug they should be getting. And we've sometimes wondered in reports as to whether or not, as increasingly we use monoclonal antibodies, which have drug targets that are typical, they may also have immune-based mechanisms of action, whether or not certain polymorphisms that exist within all of us for complement binding or for antibody binding might actually affect clinical outcome. There have been a number of proposals in the literature that, in fact, that these mutations or these variations in FC receptors can affect the binding of these drugs to the immune effector cells. So far, the studies really don't support that. But it's a novel approach, again, saying just because we're giving a new type of therapy doesn't mean the old pharmacogenomics isn't important. It's important now in a different way, something else for us to consider. And so, summing it all up, it's important to remember that the development of cancer therapies has changed dramatically in the last 2 decades as a result of these advances in molecular biology, in understanding the natural course of disease in a patient, predicting which drugs are most likely to be a benefit, helping us predict what the toxicity of a particular drug might be. These haven't caused, as a consequence, total R&D costs are up for each drug application. So defining segments of patients that are smaller but more likely to be responsive allows us to reduce the time it takes for clinical development and, as a consequence, help reduce clinical development costs. It avoids treating patients who aren't going to benefit, something that all of us want to try and use as a goal to better selecting patients. So these predictive biomarkers enable smaller trials, shorter time to benefit, and greater therapeutic effect. Crizotinib was approved from about 4 years from the time it went into drug trials, whereas for many agents it's 10 to 15 years. Vemurafenib was even less, because they knew exactly what patients they wanted to treat. And these pharmacodynamic biomarkers are needed to help us validate predictive biomarkers. I told you I'd fit Latin in here somewhere. This is from Paul Ehrlich, who's one of the great giants of medicine and therapeutics, basically saying if your drug doesn't bind to its target, there's no chance it's going to work. And that's what the FDA has said in that little asterism. We want to know, is there a dose-dependent manner for benefit and for toxicity that helps us judge how to use this in clinical medicine? And so although pharmacokinetics is not nearly as exciting, perhaps, as some of the most recent innovations like the molecular genetics, it's still a decisive factor in clinical trials. If a drug's not absorbed, if it's not predictable, if it can't be dosed properly, it's not a drug. It's just a biological observation. Pharmacogenomics can allow us to get better pharmacokinetic predictability and potentially allow us for greater safety and fewer adverse events in patients once we've selected the appropriate therapy for them. So it's just to emphasize that personalized medicine exists on multiple levels in terms of efficacy for patients, safety, selecting the appropriate dose and schedule. So, thank you.